How's everybody doing?
Good.
There is a Turnitin link on the Moodle site for turning in your paper. So if you haven't done that already, you can do that whenever. I forget what I put as the due date on it. I think it was 11/15, so that if you hadn't done it by the beginning of class today, you could still submit it and then make any changes that you need to make and still get it in. So that is that. I have names in a hopper. I'm pulling out a name, and the name I pull out is Kiara.
I had a feeling that would happen. Woohoo!
Why did you have a feeling that would happen? It was totally random.
It is totally random. I don't know. Sometimes you just get a gut feeling. So.
But anyway, now you are on the board. You will have done your penance for the year.
Perfect.
And all I can say is lead a clean life from here on out, and this won't ever happen to you again. All right, so she's going to lead discussion, and I'll be around to help her out. Give me a sec to pull up my version of the paper. So yesterday, I don't have office hours typically on Tuesday, and I wasn't actually around yesterday because I'm reviewing grants for the Herpetologist League, and those are due. And so I was at home yesterday reading grants about all kinds of different things. And one of the reasons that I read grants, that I volunteer to do that, it's a totally unpaid service that I provide, as well as some other people around the country, is I get a sense for what graduate students are putting out in terms of, it's a graduate student grant program that the Herpetologist League does. It was a fund that was set up in honor of a guy named Ernie Williams, who was a very productive herpetologist, did a lot of work in the neotropics, did a lot of work on the genus Anolis. Anyway, it's a grant program in his honor, and it's available to graduate students. And so it's nice to see the kind of quality that graduate students are putting out from places all the way from, relatively small schools with master's programs to big schools like the University of California, Berkeley, doctoral students. And anyway, I've been in the throes of that for the last couple of days, and that's coming to an end. But if you couldn't find me in my office yesterday, it's because I was home doing that. Anyway, as a result, also today during office hours, I will not be in my office. I will be at home continuing to work on those to try and get those pushed out. But if you do need me for something, by all means, drop me an email. We can either handle things via email or set up a Zoom meeting from my house. All right. Before we start, what were challenges you found with reading this paper, or were there any?
I thought the structure of the paper made it kind of difficult to read, that they didn't break up like results and discussion, because I was reading it and then all of a sudden I was like, oh, it's over. Okay.
Yeah, so what you saw was kind of a melding of methods, results, and discussion all together. And this is common at Science and Nature, the two kind of viewed as the two biggest, most prominent publishers. in the world. A number of years ago, a couple of decades ago, actually, started moving towards reducing the amount of page space that they devote to methods. And so, for example, in science, you oftentimes see the methods presented in a series of footnotes. And so it just goes from introduction to results and discussion without any methods mentioned at all. And in this case, there's kind of a seamless melding of methods, results, conclusions all together. So they present their first three, their three questions that they have. And then for each of those questions, they basically reintroduce the question. They talk about the method that they use for answering the question. They find out what, and then they discuss what the result is. So it is a different structure than perhaps you're used to in most mainstream journals where it's a little more segmented. What else?
That was
I would
kind
say,
of
like, the
mine
method
too, and
Quinn first, and then Elizabeth.
that's what I was just about to say, like the methods, kind of what Kelsey was saying, like the structure. And then when I was trying to go over the methods, they didn't seem that clear, and I had trouble like figuring out exactly what they were doing.
Mm-hmm.
So
yeah, I'll show you kind of, when we get into it, I'll show you kind of what I did with the whole thing to kind of help me with that. Elizabeth, what were you about to say?
I was going to say the exact same thing. Like the methods, they just mention whatever like theory they're using or technique they're using, and then like it's never explained beyond that. And so it was kind of difficult for me to know like what they meant by what method they were using.
Yeah. In a Nature paper, they just assume that if you're reading in Nature that you know what's going on, and if you don't, that you have ways of figuring out what's going on. So yeah. All right, Kara, you want to get us started? I realize you don't have a plan because you, for some reason, you psychically had an inkling that you would be in charge, but you probably don't have a plan. But how would you begin if anybody were starting a discussion of the paper? Go for it.
Okay, well, maybe the first thing we should talk about is their overall takeaway or a big conclusion from their paper. Just that way we can get a sense and kind of start digging in that way.
Just so you know, my effort is going to be silent during most of class today.
So they found that it did support the theory of island biogeography, but then they also talked about how human involvement also needed to be considered. That was kind of like the last bit of the paper, just the trade patterns and how that was possibly different in Cuba, I believe, where there was the trade embargo.
Yeah, I also thought that was kind of interesting because I was trying to center my paper around the 3 predictions that they made and then the 3 tests. And then once they had made their conclusions about that, they were like, well, and this thing about human. So I thought that was kind of interesting and had to structure my summary a little bit differently too. Okay.
so I also
Okay,
think
does that summarize what you just said? Okay, continue.
I was just gonna say, like with the human like activity that they talked about, they also talked about looking at like models to test the theory like through like a present day lens, because they talked about how there weren't a lot of models that were being used to test the theory, and the ones that they did use were all looking at it like in the past, and so finding new models that I think it's like the Anthropocene, like present day biogeography.
Yeah, so basically the second point here, this is point 1 of their conclusions, point 2 of their conclusions. Yeah, you have to include humans in here somewhere.
Okay. Well, now that we have those down, that's good. I talked about the 3 different predictions in their paper. Did anybody do that I'm assuming everybody did that as well. Did anybody focus on one more than the other that we want to start talking about?
before we get there, can we talk about their second paragraph?
About the different experimental methods.
Yeah, so tests of the equilibrium theory of island biogeography. ETIB I have on my board. It's the equilibrium theory of island biogeography as its formal name. So they talk about the fact that tests of this theory are lacking in many cases because of the scale that you have to do this at. And they mention one set of experiments. Somebody described those experiments to me. They're described briefly in the second paragraph.
Well, they talked about how it was hard to do these experiments because of it's hard to manipulate like experiments at such a large scale that was necessary. And so the one they were talking about was like the 6 small islands, which weren't even isolated, I don't think. And there was something about like recovery. I was a little confused on what it was, but it was the arthropod defaunation of some island. Well, I guess not islands, but 6 areas. And that
Well,
was like
they were Mangrove Islands. Yep.
Okay, but that was like the most in-depth study up to this point, but it didn't even really, it wasn't as large scale as it needed to be to look at the theory accurately. And it was also, the islands weren't isolated.
Okay, so I'm going to share my screen real quick with you. So these were studies done by E.O. Wilson, who is at Harvard, and Daniel Simberloff, who's now at Tennessee. I think he was at the University of Florida when he did this work, if I recall correctly. But if you just type Simberloff and Wilson and go to images, there are mangrove islands off the coast of Florida. Some of them are really small, some of them are larger. But the nice thing about these is they were small enough that they could go in. Can you guys see my web browser? They could go in and they could build scaffolding around these little islands, and then they hung plastic tarps over them, and then they gassed them with insecticide. And then after they gassed them with insecticide, they basically watched what happened over time. And what they saw was that over time, the insects recolonized the islands, and they recolonized in ways such that after 680, 700 or so days, that the levels on each island had come back to roughly what they were before they defaunated them. And so island size basically set the saturation point, and the rate of colonization, which was determined by distance from the mainland, essentially determined how quickly these islands returned to kind of their equilibrium number of species. And so the criticisms in this paper that they were at relatively small scale with relatively low levels of isolation are well-founded. But once again, the one really good experimental test actually bore out that the theory does work. Do you know of any other tests of this idea that have been done?
It wasn't really a test, but they did, like there was a volcanic eruption, I think, on an island called Krakatoa, I think.
Oh, yeah.
Island Reddit ecology.
Yep.
And
Krakatoa.
It wasn't a test, but they observed recolonization over time.
Yes, they did. This was an island that, what was it, 1882 or something like that? Do you recall the date?
I don't remember.
Anyway, it just erupted again and basically did the same thing over again. that basically obliterated the island. Let's go to Google Earth. I've been to Krakatoa, actually. I went to Krakatoa in 2010. It's a kind of a trippy place. Can you guys see Google Earth? Google Earth, yep, you got it.
Yep.
So Krakatoa is a volcanic cone that was once an island. It's in Indonesia. So we were talking about Indonesia just recently. So we have Sumatra, we have Java. It's in the strait between Sumatra and Java. And you can arrive in Jakarta. You can take a taxi out here to the coast and hop on a boat that seems way too small of a boat to go on a cross the ocean voyage. But we did it anyway. And you can go out to Krakatoa and visit the island. We actually climbed partway up Krakatoa while it was actively erupting. And then at the end of the day, we went and we stayed at a little camp, a wilderness camp right here on this little tiny island just off the coast of Java. But when you zoom in on Krakatoa here in the middle of the frame, you can see that this is an island named Rakata. And you'll notice about Rakata that it has this kind of curved outline here. And if you look at it carefully, you can see that it's kind of a steep cliff going down into the ocean. This is the crater that was formed by the original eruption back in the 1880s of this volcano erupting. This edge also is the edge of the cone as well. So it was this big island. It blew up out of the ocean and obliterated itself, and then started rebuilding itself from underneath the ocean. And then scientists were aware of this because you could evidently hear the eruption, audibly hear the eruption in Australia. That's how big of an eruption it was. You could hear it in mainland Australia, and seismic monitors in the United States at the time actually registered the eruption in the United States. So it was literally an eruption that was so large that it could be felt around the world. If you are in Google Earth, you can click on timeline, and you can see the island at different time periods. This is the most recent photos of Krakatoa. You can see that the outline of the island is different than it was in the past. Let's go back to 1985. And that's because it has recently obliterated itself again. So you can see the island getting bigger as it grows in time. And like I said, I was there in 2010. So in 2010, it started forming forest along one side. It continues to erupt intermittently. Like I said, we were there during a time when it was active. But then I forget if it was about 3 years ago or 5 years ago, it erupted again. And when it erupted again, once again, you can see this forest along the edge here. When it erupted again, it basically obliterated itself once again. So once again, you'd see mostly volcanic, but once again with this forest developing. And then there comes a point where oh, that's it, looking like it's fairly eruptive. And now all that forest is gone. The shape of the island has changed. And this is basically this toxic runoff that is coming out of this new crater. This new crater is filled with water because it basically totally destroyed the island and then built itself back up again from the ocean floor, and some of the seawater became trapped in it. So we have the opportunity to once again look at this thing become recolonized by animals and plants again. So this represents a kind of repeated experiment in island biogeography. And of course, the organisms that are going to colonize this are coming from the surrounding islands and are also coming from the islands of Java, Sumatra, and to a larger degree, Borneo over here. So a lot of sources for species, but they're all going to be contributing potentially organisms to Krakatoa as it recovers from its most recent eruption. So that's one of the other things that they mention. Do they mention anything else? So it's weird that they didn't mention this because this is a well-known ecological experiment. But if you go to South America in the forest north of Manaus, Brazil, you will be able to see little patches of forest that have been cut out in perfect little squares. There's 2 of them here. Let's go back in time a little bit. There's one of them here. There's another one of them here. There's a little tiny one right here. This is an experiment that is known as the biological dynamics of forest fragments. Here's one here that is connected to nearby forest. Here's a tiny one here. These biologists wanted to apply island biogeographic theory to forest fragmentation. So they went into the forests of Brazil, north of Manaus, at a time when there was still primary forest there. As population expanded out of Manaus, they wanted to set these things up far enough away that they could then have preserved little sections of forest as time goes on, and you can see the human population grow out towards them. So you can see development moving out into the forest. And because they made these things at different sizes and at times at different levels of isolation from other forest fragments, because for example, here's a mine right here. And over time, this mine has been, has had more forest clearing going on around it. So different levels of isolation as well as different sizes. And they just plunked down these plots and then watched to see what happened to species diversity inside of them after the time that they were isolated. And did they reach some equilibrium point that was indicated by not only their overall size, but also their distance from intact forest. And that's a pretty cool experiment that has been going on since the mid-1970s that has been looking at these questions of the application of island biogeography applied to conservation issues. If you're going to erupt a nature preserve, how big should that nature preserve be? How far away and isolated can it be from other similar habitats and things like this? That's an ongoing question in conservation biology in the design of nature preserves, where the goal of the nature preserve is to preserve species diversity. And so I was a little shocked when I first read this paper when it came out that they had totally ignored the biological dynamics of forest fragments experiment in South America. I would say it's because they're unaware of it, but I can't believe that Jonathan Losos is unaware of it. But anyway, there are more examples of tests than they mention in this paper. There just aren't a ton more examples of tests of the idea. Now I'm going to go back to shutting up again. So Kiara, you were talking about the 3 questions that they had.
Yes, and does anybody want to start on one more than another?
Let's just start on the first one that they mentioned was have exotic anoles established on banks furthest from the area set saturation point.
Okay, so does anybody want to state first maybe what they found and then the methods that they used to go about seeing about this prediction?
So I think that they were using the saturation points of the native anoles, and then it seemed like they were doing a lot of estimating because they estimated the bank saturation. And so I know that they used regression and statistics to be able to check their work, but I thought it was funny just to comment that they openly said a lot in this paper they were estimating.
So first of all, everything that you do in science is estimating, because you hardly ever have access to the actual population of observations that you're interested in. So everything is an estimate. The mean is an estimate of the population mean. The sample mean is an estimate of the population mean. And so there's nothing inherently wrong with estimating. What they're doing in this case is they're basically using residuals, which is something that you learned about when you were first years and you probably don't remember it. But as an example, let's take something that's easy to think about. The height of individuals and the speed of individuals. It makes sense that taller people have longer strides, and longer strides should make you faster because each stride covers more distance. So you might expect from short individuals to tall individuals, you might expect speed from slow to fast to increase, such that there is a general relationship between height and speed. But at a given height, you might have some individuals who are faster than other individuals because of reasons unrelated to height. So at this particular height, this individual would represent a slow individual. This individual would represent a fast individual because at that height, this person falls below the line, which is kind of the height corrected average speed. This individual falls above the line. This distance from the predicted line to the actual data point is referred to as the residual. Now, this individual here is slower than this individual, but its residual speed is faster, is bigger than this individual, which means that for that stride length that they have, they're running faster than this individual, even though this individual's raw speed is faster. And so what they're doing in this is that they're taking the relationship, for example, between island size, which should be the ultimate determiner of how many species can live on an island. They're regressing island size versus species number, species richness. And at a given island size, some population or some islands are going to have lower number of species than you would expect. Other ones would have higher than you would expect. And those expectations are determined by those residuals. And so you can separate islands into islands closer to saturation or further away from saturation, depending on whether they have positive residuals, they have more species than is predicted by the regression versus fewer species than are predicted by the regression. So they're not really predicting what the actual saturation value is. They're kind of using the regression to say, well, what's the kind of saturation that you should be expected at a given island size? Are you below what you might expect, or are you above what might you expect? And if you're above, then you're closer to saturation than if you're below the regression line. And so that's why they're using residuals. Does that help with understanding what they're doing methodologically? Okay. All right, continue. Rose, you're doing good.
So what they found from this question was that the exotic anoles established themselves on the banks that lacked the native ones, and this was aligned with their prediction that those impoverished banks would allow those exotic ones to have more opportunity to become established and thrive.
Continue.
Well, that's just what that question was.
So among the islands, they had some islands that they said were more saturated and other islands that were more impoverished. Generally, the impoverished islands were what type of island? There's a particular set of descriptors that kind of define whether or not an island is going to be impoverished or not. And they talk about this more later in the paper. What are the two processes that add species to islands?
Colonization and like speciation. So like impoverished banks or islands should be islands that are large, but then like are set way below their saturation point because they're further isolated.
Okay, so impoverishment can be due to isolation. They're just really far away, such that there hasn't been enough geological time for enough colonists to have reached there. But what's the other thing that can lead to impoverishment? So you mentioned large islands that are super isolated. So extinction occurs on small islands, but what else doesn't occur on small islands?
Speciation.
Yeah, speciation.
Well, yeah.
So they talk in the paper about many of these impoverished islands actually end up being islands that are so small that in situ speciation doesn't occur. And so they don't have their saturation, their equilibrium number of species, not because they're not getting colonists, but they're also not getting in situ speciation. And so the impoverished islands also tend to also be the smaller islands within the Caribbean. And that's something to always keep in the back of your mind as you go through the rest of the paper.
Okay, based on
Rose, sum up the overall conclusion regarding this question again, since we've deviated somewhat.
That the impoverished banks are where the exotic anoles establish themselves.
Okay, that's pretty much it. Continue, Kiara.
I was going to ask if we agreed or disagreed, but I know we said we didn't talk and know a lot about the methods. So does anybody want to take a stab at why they would agree or disagree with this conclusion?
I would agree with it because it makes logical sense that if they didn't have as much competition, that it would allow them the most opportunity to become established and thrive.
Okay, does anybody else have anything to say about that one? Otherwise, we can move to the next one. No? Okay. All right, so the next one talked about the anole colonizations and how they could have diminished the past signal of speciation on bank richness. Did anybody want to talk specifically about that one?
I mean, to break the silence, I'll say that I did have trouble with this second question, and I thought that how they went about answering their own question was confusing, kind of, and I know the reasoning behind why they didn't explain as much as they should have, like Paul said at the beginning of class, but I think I'm still not completely sure how they answered their own question.
Yeah, the reason nobody wants to talk about this one is this is the meatiest of the three questions, and it's also the more complicated one to answer. So somebody else take a stab at it.
So I guess as I approached this question, I was kind of looking at, like we kind of already talked about how as islands become more isolated, diversity decreases. And as a result, like speciation kind of also decreases so that there's less diversity on more isolated and smaller islands. And so, like, as I kind of walked through it, because there's more of this human mediated colonization, it's increasing the phylogenetic variation on these more isolated islands. Which is having like an effect. And I'll skip that part. And then because there's more colonization as well on larger islands, it means that those colonists are fulfilling more quickly any like available niches. So like speciation like would take longer and would be less likely to happen.
So does island size influence colonization rate, or does level of isolation increase colonization rate? Because you just mentioned size and colonization rate together.
Like, yes, like size affects. Colonization rate, like in terms of let me think about this. Sorry.
What's that? You're breaking up a little bit.
Sorry, just give me a second to think about it. Like,
okay. Everybody look at the last full paragraph on page 543. Second column has the increased colonization obscured the past effect of speciation. The first thing that they deal with is isolation. When you're isolated, when a colonist does get there, species diversity increases due to what process in isolated islands? If you're isolated, does species richness increase because of a lot of colonization, or does it increase because of in situ speciation after a colonist gets there?
In situ speciation.
Yeah, so if you're an isolated island, you get more species by that species that arrives, then evolving into a bunch of different species. And so they mentioned in this paragraph, phylogenetic diversity isolation relationship. What's the relationship between the isolation of an island and how much phylogenetic diversity is on that island? And you should expect, in a world free of humans, what should you expect? Should you expect a lot of phylogenetic diversity on isolated islands, or should you expect limited phylogenetic diversity on isolated islands?
Limited.
Limited, why? Was that Kelsey?
Yeah. well, if there's not like humans moving anything around, then the speciation events are going to be like naturally occurring, or like natural like immigration and immigration. So they talked about like how these lizards couldn't do like aquatic dispersal because they can't travel in the water. So that was limiting island hopping. But then when the humans were coming in and transporting them, then they were able to, like the phylogenetically different lizards were able to move from island to island as a result of human aid.
Yeah, so when you look at figure 1, you see that many of these clades, you know, all the descendants of a common ancestor happen to also all live on the same island or the same island bank. And so all of the species that live on an island are closely related to one another, so phylogenetic diversity is low. And this just happens to be especially true on isolated islands rather than non-isolated islands. If you're closer to a mainland source of species, the odds are that you're getting more colonization events, and so then you get more of these individual clades developing on a given island. And this is the case with Cuba. Cuba is very close to Florida, and so on Cuba you have a bunch of different clades that come. So if you look at figure 1, Cuba is the second from the top island, and you can see that there's species on Cuba from lots of different clades throughout this phylogenetic tree on the left-hand side. And that's because they're just getting lots of independent colonization events. And so the phylogenetic diversity of Cuba is high, not because it's a big island, although that certainly helps, but it's high because it's a close island to a continental source pool of species. So in this case, it's physical isolation that matters from the standpoint of phylogenetic diversity. So they expected phylogenetic diversity to have an inverse relationship with isolation. The more isolated you are, the less diversity you have. More isolated, less diversity. So that negative relationship. So then what happens when you start including exotic species into the mix?
The phylogeny was more random.
Yeah, and what happened to this relationship between phylogenetic diversity and isolation?
I think they said like the relationship like basically became non-existent once like exotic anoles were there.
Yeah, did you guys look at the extended data figures as you were reading to kind of help you through this? What extended data figure is this question illustrated by?
Three.
I think you're 3.
Yep, figure 3. And so it shows past phylogenetic diversity and present phylogenetic diversity. And with the past phylogenetic diversity versus isolation, there's a negative relationship. And then you lose that relationship largely because the upper right-hand corner of this figure gets filled in by a bunch of these isolated islands having way more species than they had before. So you can look at where points are moving in this figure. So you see the, let me do this. So that relationship is destroyed by that area at the upper right-hand corner that in the past didn't have any islands in it because they were really isolated and had relatively few species. And when you start including introduced species into those islands, then you get that corner populating, which then raises raises the slope of that line, making it no longer a negative relationship, but just a flat relationship. Continue, Kiara. So that was the first line of evidence. What was their second line of evidence for this question?
They talked about Nc2 speciation.
Yeah, go with that.
Okay. Well, they looked at the nonlinear function, which corresponded with Nc2 speciation, and they basically found that SAR exhibited the nonlinearity, but now in present day that the pattern of that is gone, and it's more of a linear model now than nonlinear.
And where is this found in the paper in terms of a figure?
I think it's 2A.
Yeah, so I think in the figure, like in A, they showed like the two parts. So like at like that point, like it started increasing dramatically, but then when it gets to B, like the present day, it, like you said, it's linear, and it's like basically the two-part species area relationship was like eliminated.
Yeah, it's this section right here. And I've circled the areas where you can see that islands shift from past to present in terms of their species number. And when that shift happens, that inflection point disappears. And the small island effect is commonplace in biogeography. In the Caribbean, I did a study a number of years ago with spider species, and that inflection point appears for spider species as well. Below a certain threshold size, basically all of the islands look very similar to one another in terms of species richness, simply because they are not big enough to support in situ speciation. So all of the species that live on those islands are species that also live on other islands in the Caribbean as well. And it's only when you get an island big enough to support in situ speciation that you then begin to see species richness increasing at a rapid rate. So the small island effect is something that happens over and over again, regardless of the kind of taxa that you're looking at. It works for spiders, it works for anole lizards, it works for orchids in the plant world. So it's a common idea in the Caribbean and in island biogeography in general. In the literature, it's called the small island effect, essentially. Continue, Kiara.
Okay, does anybody else have anything to say about this one? I know it was hard for some of us to understand, myself included. No? Okay. Do we want to talk about the third one now, about how the anole SAR strengthened and the SIR weakened? Or maybe I should ask why. Anybody know why?
Because the exotic species were colonizing the impoverished banks, like the increase in the number of species or species richness was moving it towards like the species saturation point or whatever that term was. And so that would strengthen the SAR.
Yep, exactly. So whenever I see SAR and SIR, I have to just remind myself that it's the species area relationship and the species isolation relationship. So as area goes up, what should happen to species number?
Should it increase?
Yep. And so, like Kelsey said, as you get more exotic species, that relationship just gets stronger. As isolation increases, what happens to species number?
Decreases.
It increases or decreases?
Decreases, right?
Decreases, yeah. So if you have exotics coming in, what should you expect to see? As isolation increases, what should you expect to happen to species number?
Species number would decrease if isolation is increasing.
Yeah, sure. And so
SIR,
did they find that weakened or strengthened over time?
it weakened.
Where do you find this information in graphical form?
I think it's still in figure 2.
Yeah, I think it's 2C and 2D.
Yeah.
Yep. So when you look at figure 2, it's good to decompose figures sometimes. So what are the blue and the red dots referring to?
The red dots are banks with Nc2. Oh, sorry. Speciation, and the blue are dots without, I think.
Okay, so red is with speciation, blue is without speciation. What's the difference between open and closed circles?
The open should just be native species, representing like the past situation, and the filled should be exotic species.
Yep. And so when you look at C and D, the red line is showing the relationship between isolation and species richness for islands that have in situ speciation. And those lines don't really look very different from one another, do they? What happens though to the blue line between figure C and D of figure 2?
It inverses?
Yeah, the slope goes from a negative slope to either a flat slope or maybe even a slightly positive slope. I don't remember the details, but I think it was a slope not significantly different from 0. And once again, you can see which islands it is that are responsible for that, because it's all those dark blue ones that now have higher species diversity than they had in the previous data, which is figure C. So that's what's destroying the species isolation relationship, is these islands without in situ speciation are gaining exotic species, and that's even happening on really isolated islands, the islands to the far right-hand side of that figure. As you're going through a paper like this, it's oftentimes useful that when the authors say something verbally and then refer to a figure, that you go to that figure and try and figure out, okay, how does this figure illustrate what the authors just claimed? And it will help you in developing skills of figuring out figures and what figures mean. But oftentimes seeing the figure will help reinforce what the authors just said. And this paper is especially, that idea is especially true in this paper. Continue, Kiara.
Okay, so we've talked about all three of them, and they basically just support the theory that ETIB works. So I guess we could talk about now how important it is to include human activity. Does anybody want to talk about basically the different trends that they saw within the exotic anoles and different areas of time?
I thought it was interesting that they pulled up that figure. I think it's figure 3, where they basically showed how the anole population, exotic anole populations changed like post World War II and post the Cold War, and how that was like also associated with increased shipping worldwide. I just thought that was interesting. Like,
yeah, for me, one of the cooler aspects of this paper is the stuff that comes here at the end of the paper. And so Elizabeth was pointing out the graph in figure 3, where it's showing that over time you get more species on these islands, but it's not linear. It comes in spurts, and these spurts are keyed to economic events and geopolitical events. World War II was one of them, and the time immediately after World War II. But then at the end of the Cold War, when you had more global trade going on, because you no longer had the United States and Russia kind of dividing the world into two sets of allies, allies of Russia and allies of the United States. And that line after the end of the Cold War, that thing just shoots up. It's fairly impressive. Continue on this vein.
I'm trying to think of a question. Hold on.
So here's the question I would ask. So for all of these analyses that they've been doing up to this point in the paper, what's the measure of isolation that they have been using?
Haven't they been measuring species richness?
Well, what's the measure of isolation that they've been using?
I think they use 3 as a way of like accounting for distance of like a singular bank from other banks, and then from Cuba and northern South America, as that's where like they posited that the lizards originated.
Yeah,
so, like,
so there's only one species of anole that is native to the United States. So any anoles are going to be coming from South America or Central America. So extended data table 4 basically shows the places where they're thinking about in terms of isolation, distance from Cuba and distance from South America and the combined distance, as well as distances between banks. So when I ask what has isolation been based on up to this point in the paper, it's basically based on what? It's just physical distance. Now, what shift do they make here at the end of the paper? isolation is now going to be based on what?
I mean, like economic activity, shipping. stuff
They
like that.
describe it as economic, but it's
Yeah.
based on the number of ships at each port.
Yeah, Kelsey and Elizabeth are both right. It's a degree of economic isolation. In this case, what's the most economic isolation, the most economically isolated island in the Caribbean?
At the time, I think it was Cuba.
At the time it was
Because
Cuba,
it
and it
was
still
like
is
the
Cuba.
US.
Where is Cuba located?
Now off the coast of Florida.
Yeah, so it is really the least geographically isolated in terms of distance from a mainland source of species. It's really not very isolated at all. But economically, it's among the most isolated because we don't do trade with Cuba, and as a result, most of our allies don't do trade with Cuba. And so as a result, it's very much economically isolated. Where do you find this illustrated best?
They use the tables, but I personally thought the tables kind of were hard to interpret.
Okay, so what's easier to interpret?
Figure 3.
In the extended figures?
Oh,
Not figure 3 in the paper.
figure
itself, I don't
4.
think. Figure 4 in the extended figures. Go to those guys. What do you see in those figures? Anybody?
You see the economic isolation area relationship across the different banks, and then they measured the residuals of the different ones.
Yeah, and where does Cuba appear?
Cuba is up here.
It is way up there, and where is it in the residuals?
Isn't it the largest residual?
Oh, largest by a lot, like twice as far from the predicted line as all the other islands in the Caribbean. Who's the least economically isolated island in the Caribbean?
Is it Puerto Rico?
Oh yeah, it's Puerto Rico. Why is that?
Because it's like receiving the most like US involvement.
Oh my gosh, it certainly is. So when you look at the Caribbean, you see Florida sticking down, you see Cuba here, you see Hispaniola here, and you see Puerto Rico way over here. Yet Puerto Rico has less isolation than Cuba or Hispaniola, even though Cuba and Hispaniola are closer to the United States. And it's because Puerto Rico is a free associated state associated with the United States. It's not a state, but it's kind of sort of like a state. Guess what just happened this past week in the US House of Representatives? With regards to Puerto Rico. Oh, you guys don't follow Puerto Rican politics in the House of Representatives? Come on. Last year, there was a referendum in Puerto Rico. They have these referendums in Puerto Rico periodically about whether or not Puerto Rico should try and become an independent nation, independent of the United States, whether they should remain in their current status, which is technically a free associated state in alignment with the United States, or whether they should apply for statehood. And for the first time in the history of Puerto Rican referendums last year, the Puerto Ricans voted 53% in favor and 47% against applying for statehood. So it's the first time that statehood has won in a referendum. Typically what happens in referendums in Puerto Rico is 48% of the population want statehood, 48% of the population want things to stay the same, and about 2% want Puerto Rico to become its own independent nation. So it's never, statehood has never gotten a majority vote in Puerto Rico. And now that they have gotten a majority vote in favor of statehood, they have been pressuring mainly Democratic leaders in the House of Representatives to put forth a formal bill that will start the process of moving Puerto Rico from being a free associated state to being an actual state. And Democrats want this because chances are that would give the Democrats 2 Democratic senators in the Senate, and it would give them 4 Democratic representatives in the House of Representatives, and Puerto Ricans would now be able to vote for president. And if it was a state, those state electors would go probably in a Democratic way. And Republicans don't want that to happen because they want to keep control of the House and the Senate if they can possibly regain control. So anyway, we are very closely associated with Puerto Rico because it's basically part of the United States, and may become more of a part of the United States in the future. Cuba has been under embargo by the United States since the Cuban Missile Crisis back in the 1960s because of its close association with Russia. And I don't know that that's going to change anytime soon. So economic isolation is very different from physical isolation because of geopolitics. And what did they find when they looked at economic isolation relative to species richness?
Since there was a negative function of economic isolation and annual richness.
Yeah, so the species isolation relationship, which we should expect to be negative, in the past data looking at native species, it was negative. In the present, it was not different from 0. So this is based on physical distance as the measure of isolation. So when you just look at physical distance, the movement of exotic species around the Caribbean has gotten rid of this negative species isolation relationship. But when you shift from looking at modern data and using physical distance to using economic isolation as your measure of isolation, and evaluate present data, what happens to that relationship. Is it still 0 or is it positive or negative?
Negative.
Yeah, it's come back to negative. So in this case, the economics of the situation actually reflect what used to be the situation with geographic isolation. So knowing something about the activities of humans sheds light on the biogeographic patterns that you see in the Caribbean, at least among the knolls. And we've now killed more than an hour on this. So that's it. Quiz on, or a test on Friday. It's going to be similar to the last test, and that it's going to be mostly about reptile families and a little bit of stuff about biogeography here tacked onto the end. Let me know if you need anything between now and Friday. Friday, we'll do the same thing that we did last time. I'll post a PDF as well as a Word version of the exam on the Moodle site, so you can have access to both of those, whichever way works best with whatever interface you're using to take the test. Email me with questions between now and then if you have questions about the test. But it should be a very similar format to the last one. All right, I'll see you guys in lab tomorrow.